The broadcast is now starting. All attendees are in listen-only mode.
Okay, so good afternoon and good morning, wherever you are in the world. This is Richard Ellis. I'm Business Development Manager at the Phenotypic Research Institute, and we're here today to do this webinar with our friends in Canada, DNA Genotek, who are also members of the PRI. So it's just a quick introduction. The PRI was formed 7 years ago to clear up the regulatory pathways around live biotherapeutic products in Europe. Since then, we've become a real center, a network for all the different actors involved in the human microbiome drug development supply chain. If anybody's interested, then please feel free to get in touch with us, Richard at pharmabic.org. So I'm going to now hand you all over to Aaron Del Duca, Vice President of Technology of Microbiome Program at DNA Genotek, who's going to take you away with this very interesting webinar about curating massive amounts of biological material and phenotypic data for efficient and reproducible microbiome discovery. So, Aaron, we're going to put you on, and over to you.
Thanks, Richard. I'm really glad to be with everybody this morning or this afternoon. I wanted to take an opportunity to share a little bit of what's cooking in DNA Genotek's R&D kitchen, so to speak. We'll talk a little bit about who we are and what we do, but wanted to lend some insights to the folks who are, on one hand, looking to start up studies, get into study design, make sure that they're maximizing their research investments, and as well provide some insights and a heads up for some interesting collaborations for those of you who are looking to consume or source microbiome information from studies that are happening in academia. So we'll kind of try and present some interesting perspectives on both sides of the table. So DNA Genotek has been in the medical device space for the better part of about 13 years. What we do is we have a really deep understanding of genomics and molecular biology, and we have a really good understanding of what it takes to process things at high throughput in a regulated environment and in high throughput molecular labs. We also have a really good understanding of human factors and device design. And so we combine this know-how with our proprietary platforms for stabilizing a variety of molecules, whether it be DNA, RNA, or metabolites. And we combine all this know-how into medical devices to allow discovery at scale in a reproducible and quality controlled fashion. So on the bottom, I'm just calling out a few of the customers that we've been really fortunate to work with over the last few years. On the genomic side of our business, we've been with 23andMe since their first sample has been collected and have supported them for the last 10 or so years. So we really do understand scale. Same for Helix. And on the microbiome side, we're really fortunate to be working with some of the brightest minds in the field, providing them the picks and shovels, literally and figuratively, so that the tools of the trade, so to speak, to make sure that what they're capturing really does represent that in vivo condition that they want to observe and learn about. So toward the end of the presentation, like I mentioned, I'll call out two collaborations that DNA Genotek has recently embarked on, one with the T.H. Chan School of Public Health at Harvard, and another one with Janssen, so stay tuned for that. It's definitely an exciting time to be looking into the microbiome. Funding availability for research is growing by leaps and bounds. And if you take a look at where that research funding is going, a fairly healthy portion of it is directed at human and clinical discovery efforts. By our latest count, there's about 100 or so therapeutics in clinical development around the world, and quite a bit of applicability and relevance to non-human indications of the microbiome as well, whether that be environmental or agricultural or what have you. So we're seeing tremendous growth, and I think that's one because it's trendy and that happens in research, but also because we're answering, or we're able to answer, really important questions and provide important new insights with this kind of exploration. And if that wasn't exciting enough for you, then you just had to go to the American Society of Human Genetics meeting in Orlando a couple weeks ago, where Francis Collins and Bill Gates were having a fireside chat about this very topic. And Dr. Collins asked Bill Gates, you know, in the world of genomics, what are you most excited about? And his answer, I think, dropped a lot of jaws, and his response was the microbiome. So I might as well stop there. But it's certainly an important time to be thinking about how to make sure we're doing the best we can with these microbiome research and discovery initiatives that we're pursuing. So I kind of see us at an inflection point. To date, we've had, you know, some extremely bright minds showing us the way, coming up with proofs of concept, you know, really exciting and novel studies that are kind of giving us hypotheses and ideas to sort of figure out. But some of the challenges there are, you know, when you're doing a proof of concept, it's often underpowered and sometimes difficult to reproduce. And so if we're going to get through this inflection point, really the best way through it is to focus on execution. And so by definition, if we're able to, you know, do that, we can transcend novelty and get into utility. And to do that, we have to have very rigorous statistical methods to make sure that what we think is true is actually verified to be true. We have to run things in very quality controlled ways so that they're reproducible. And the thing that I really want to focus on here is that when we do this, we create leverageable assets, things that we can go back to, things that we can aggregate and heap more on top of, and to give us a refined and an improved perspective over time, instead of these just one-off studies that are sort of islands unto themselves. So if we take a look at something that we're really familiar with and then sort of compare that to this new frontier, you know, we've got a very mature and very widespread infrastructure to help us understand genomes, right? There's tons of sequencing capacity out there. And when we take a look at the human genome, we're looking at one species with about 23,000 genes that encodes roughly 10 million SNPs. And in any given study, all of your study subjects will share about 99.5% of their genomes. And it's more or less static, right? From the time we're born to the time we pass on. And everybody accepts that this is definitely a big data and a big biology challenge. But when we look at the other side and think about, you know, metagenomics in this same context, in any one study subject, you're looking at potentially 1,000 species that encode 10 million genes. And within a study group, roughly about 30% of the gut microbes would be shared among those study participants. And, you know, to further complicate things, we have this dynamic nature to the microbiome where things change even daily, but certainly seasonally and as we age. So when we think about, you know, designing studies to capture all of this variation, there has to be a really serious consideration of how to power these things and how to make sure that we're getting the most out of our research investments. So let's take a look at the generalized metagenomics workflow, super basic, but we're taking a look at you know, stool as a sort of a high watermark for biological sample complexity, but this could be generalized to any kind of microbiome that you might be interested in sampling. So we're out there in the field, in the wild, we're collecting material from our study participants. We have to make sure it gets back to the lab. We do some extraction or processing on it, and then we take a look and attempt to do some quantitative analysis with sequencing. On the left-hand side, what I've kind of pointed out here is a really just a graphical representation of what we're looking at, a chart of who's there, how many of each are there. And the thing about metagenomics, and especially working with stool, is that every sample will provide you a profile. You'll always get an answer. But the question is it actually a very good representation of what was happening in that study subject at that point in time? Or is something happening to that sample where I'm looking at some artifacts or noise instead of that authentic profile of that study subject? And what we do at DNA Genotek in our R&D program is we sort of peel back the layers on this process. And there's a lot going on in this chart here, but really it's sort of a quantitative assessment of the sources of variation from sampling through to sequencing. And what we've done here in this experiment, in collaboration with some folks at the National Institutes of Standards and Technology in the US, is we've taken 30 donors and we've collected samples, both fresh and immediately processed, so it's our idealized ground truth material. And then we've taken samples and we've stabilized them and we've held them over time for different time periods. We've taken that material, we extract it, and then we sequence it. And we do everything in technical replicates. So we have triplicates of all the things that we're taking a look at here. And so if we walk from right to left across these box plots, you'll see that, as I mentioned before, if you look at the amount of variation between study subjects, you'll see that they have about 30% of their microbes in common. And when you look at the y-axis here, we're looking at Bray-Curtis dissimilarity. So two perfectly matched identical samples have a Bray-Curtis dissimilarity of 0. But two study subjects who share maybe 30% of their gut microbes have a Bray-Curtis dissimilarity score of about 0.75. So that's really high. The next box plot, what we're looking at is what happens when you compare a fresh sample to one that's been held at ambient temperature without any stabilization or any protection for 5 days. And what you can see is that among the 30 donors that we've sampled, some change a modest amount and some change an incredibly large amount. It's sort of difficult to measure how much things change, but all these things change to a material extent, enough to really introduce a tremendous amount of noise into any study. The next box plot over, we're looking at comparing 3 different extraction methods to one another. And this has been sort of pointed out in the literature over the last few years, is that you really do need to pick an extraction method and stick with it, because changing will introduce a lot of variation, as you can see in that box plot there. The next one over, we're taking a look at technical replicates of extraction. So sticking to one kit, starting with the same biological material and extracting it 3 times, you'll see that they're nicely reproducible. So there are robust methods out there that you can trust. And then if you take that biological material and you make your libraries, and then take those libraries and then sequence them on 3 different runs and compare those runs together, you'll see that the sequencing instruments are also very robust and very repeatable. So maybe the surprise on this chart is that the effect of stabilization, or the lack thereof, is actually significantly larger than the effect of changing your extraction method mid-study. And this is the problem that DNA Genotek set out to solve in creating our OmniGene gut device, that little tube that I've depicted on the chart here. And so what we're showing is, in the purple box plot at the bottom, if samples are properly stabilized and you take a look at these fresh, immediately collected and processed idealized samples and compare those to samples that have been stabilized in OmniGene, the differences are extremely small. Almost all of that noise goes away. And so we'll take a look at what that means from a study design and an execution perspective in just a second. So if you don't want to take my word for it, then I would encourage you to take a look in the literature and see other folks who've sort of asked that same question. How much noise is too much? And how do these other sort of homebrew stabilization methods work out there? And so I'm just kind of pointing out a few papers that have been published in the last little while who've asked that question. And I've taken that same Bray-Curtis dissimilarity metric, and I've just kind of rotated the axis 90 degrees. And that gray box plot you're seeing is the same one that we saw in the last chart that shows the effect of no stabilization. So the amount of noise that you're seeing in samples that have been left at ambient temperature without any protection for 5 days, right? So you're seeing from 0.25 to 0.75, which is quite a lot of noise. It's in that red zone. the OmniGene gut samples that were collected and assayed in these papers appear on the purple dots, close to 0 in the green zone. So they're quite stable and trustworthy. And I'll overlay a few other dots from other studies showing the performance of other homebrew methods or catalog buffers that are available. And what you'll see is that most of them kind of overlay right on top of that box plot, which is an unstabilized sample. So a lot of these homebrew methods provide no protection or any measurable benefit. Here's another special example I wanted to show you where Rob Knight collaborated with us on one of these studies. And he included a homebrew method in one study in 2015 and again in 2016. The difference between the results is that in the 2015 study, he used field collected samples, and in the 2016 study, he used lab simulated samples. So all of the work was done at the bench instead of collecting real samples out in the wild. And what that shows you is that you can't underestimate the effect of the donor, that study subject, and how he or she interacts with the material and whether or not they've done this before. That can have a tremendous influence on data quality and sample quality. So if we take a look at some of the comments that were written up in these papers, it kind of reflects well, and it kind of goes to show that what we've put into the OmniGene technology is pretty trustworthy, and it's something that you can standardize on. That's, I think, the most important thing. So with our collaborators, we've kind of taken this to the next step. We really wanted to understand if we could further quantitate this and give sampling some kind of a quality score to help people design their studies. And so we worked with some statisticians at the National Cancer Institute to create, or to look at it through the statistical lens and come up with these scores. And I'll just point you to the purple box on the right-hand side of the slide and the yellow box, and we'll take a look at that in a bit more detail here. So if you compare, again, those fresh, those idealized samples to ones that have been stored for any length of time in a stabilized device, you'll be able to create another metric of similarity or quality. The nice thing about this statistic is called the intra-class correlation coefficient, or ICC, is that it sort of relates very, very simply to N, or the study's size, the number of subjects that you need in order to achieve the appropriate level of discovery power. So if we take a look at the literature, there's a great paper by Yaron Reyes who talks about study design and as it relates to the gut-brain axis. It's a really popular and interesting area for people to be looking at microbiome connections these days. And he goes to illustrate an example where you would need roughly 500 cases and 500 controls to achieve 95% power in this hypothetical study that he's discussing. And so when you're doing your power calculations, most people assume that a study subject provides 1 unit worth of data. But what we've come to realize is that the sample quality really does influence materially the output of each study subject in that project. And so what we do is we simply divide this ICC, or this intra-class correlation coefficient, or this quality score, by the theoretical N you need to include in your study to achieve study power, and you'll actually come up with the actual number of study subjects that you'll need to achieve that study power. And on the right-hand side, you'll see that with OmniGene Gut, it's very robust. Stabilized samples are almost indistinguishable from freshly collected, immediately processed, idealized samples. Whereas these other homebrew methods, catalog buffers, RNA-later, etc., have much lower quality scores, and as a result, require you to enroll significantly higher numbers of study subjects to achieve that same discovery power. Again, if you take a look at the literature, there's a great paper by Emmanuel and Lichter that show, on average, the non-clinical cost per patient in an interventional trial, excluding the intervention itself. So this is just like onboarding, consent, maintaining an engagement, IRB submissions and so forth, is about $1,000 to $2,000 per patient. So when you think about the unit economics of that, making sure that you're wringing the sponge out and getting the best quality samples you possibly can will more than pay for itself many, many times over in operational efficiencies and improved discovery power. So that's sort of the really technical, what's going on inside the tube scenario that a lot of people may or may not appreciate. The human factors, the compliance, the usability of the device and how it's designed, I think is a lot more intuitive. But we still wanted to take as quantitative an approach to understanding usability as we did with biospecimen quality and discovery power. So what we did was collaborate with 4 institutes, Project ARC, the Crohn's and Colitis Foundation of Canada, and then Oregon Health and Science University. And we wanted to take a look at a very broad set of populations. So we took a look at healthy, naive donors, naive meaning they'd never collected a stool sample before, an elderly population of men who are 75 years or older. This is in conjunction with the Mr. Ross cohort, a study of osteoporosis and the microbiome. We took a look at mother-infant pairs and folks that had Crohn's or ulcerative colitis and provided very challenging, very liquidy stools. If you're familiar with the Bristol scale, it's about type 6 or type 7. And so what we did was we had each of these study coordinators outside of DNA Genotek monitor, how long it took to get the samples back, what proportion of samples that were sent out actually made it back. Of those samples that were returned, how many of those produced good quality results? And then in a survey, we had each of those study coordinators ask their study subjects to rate the ease of use of that sample collection modality. And so you'll see, and this was a really huge surprise to us, the sample return rates were, between 70 and 90%. And to put that into context, compliance for a phlebotomy or a blood draw is around 40 to 50%. And so we scratched our heads a bit on this, and we kind of summarized that, because you're able to send this to the study subject's home and not, make them lose time or make an extra appointment and go to a clinic and get a blood draw, that they're actually fairly motivated to provide you a sample if they have any interest in your study. So if they're interested in your study, they're very, very, very motivated to provide a sample this way. And then of the stuff that was returned, pretty much everything worked well. And again, surprisingly, folks really didn't seem to have much challenge doing this properly. That's really important when you think about microbiome studies, because to really have a good chance at understanding that, cause and effect, we need to look at study subjects longitudinally. And it's okay to get them to do something once, but it's a little tricky to get them to do something multiple times. So this human factor, this device design and interoperability really does influence time and effort that it would take to recruit and sample from an appropriate number of study subjects. So when you get this right, when you understand biospecimen quality, usability, how to do things at scale in a really reproducible way, we start to get back to the concept of creating leverageable assets. And so DNA Genotek was selected by Harvard and the T.H. Chan School of Public Health to create a sampling solution. So you'll see a sort of a graphical representation of that on the right-hand side. For a resampling of the Harvard Nurses Health Study, where 275,000 participants have been enrolled and have been monitored for a very long time, generations. They have an incredible amount of phenotypic and metadata amassed from these study subjects. And so now they'll be going out and resampling them for the gut and oral microbiome. So they'll look at 25,000 participants and collect approximately 75,000 samples starting early next year. So we're really proud to be working with them, and I'm excited to share this with this group because not everyone is really interested or motivated to create their own study group. They're looking for ready-made, biobanks or data repositories, and I think this is a very fine example of a leverageable asset that can really go a long way to, help us discover and create powerful associations. As sort of a byproduct of this collaboration, we had a great opportunity to work with Hamilton Robotics to configure liquid handling instrumentation as well as an archive for a biorepository. So we now have a workflow that anyone can use to process tens of thousands of stool samples in an automated, hands-off kind of scalable way. So if that sort of speaks to you, feel free to ping me after, and we can figure out how to get you in touch with the folks in Hamilton. And the second collaboration that I wanted to share is if you're more interested in creating your own study group or running your own clinical trials and are thinking about setting up a lab, we've set up a collaboration with Janssen, the BioCollective, and DNA Nexus to provide the materials that are required for you to validate your workflows. So what we've done is worked with the BioCollective to identify and collect stool from about 25 different donors across 5 interesting phenotypes and create a very large quantity of actual human stool that we've characterized and stabilized, so that we now have thousands of aliquots of homogenized stool. Each one is identical to the other, and we can send this out to anyone, anywhere, and you can compare, your assessment of that sample to a ground truth. And that way we can kind of learn and improve together, so we can understand how, your pipeline and your workflows might be slightly different from the ground truth set, and we can kind of work together to optimize and make everything as representative of that in vivo state, that moment of collection, as possible. So that's something that we're really excited to be participating in, and this is open to anyone to join in as well. So if you're not interested in setting up your own lab, that's something that DNA Genotek would be very keen to provide for you. In addition to the medical devices and stabilization tools, we also have a services lab and what we call sort of microbiome on demand. The idea there is we're sort of the lab behind the curtain that allows a few direct-to-consumer companies and quite a few sponsors of clinical trials to reach out to anyone, anywhere, provide them the right sort of tools, documentation, etc., and in a platform agnostic way allow them to not just sample biological material, but capture metadata, lifestyle information, other factors that would be really important to include in the analysis, either on their smartphone or on a tablet or whatever. We can kind of grab that, bring that into the cloud in a private and secure way, create sort of a two-way communication or a dialogue between the study sponsor and the study participant. Because I think the only way forward, especially with longitudinal studies where we need engagement over a prolonged period, is to provide something that motivates that study subject to stay tuned in. And maybe that's trend level information that we send back to the study participants. Maybe it's something unique about their own profiles that keeps them encouraged and motivated to keep sharing data with the study sponsor. But that's something that we can enable. And then from the wet lab processes, we can take the omics data. From the in silico side, we can collate all that metadata and lifestyle information and then we can work with you to do some predictive analytics and discovery projects that way as well. So that's available if you're interested in working with us to sort of carry out clinical trials and discovery projects together. And with that, I'll sort of leave you with a quote from one of my heroes, Richard Feynman, who's a Nobel laureate in quantum mechanics. And the first principle is that we must not fool ourselves, and we are the easiest people to fool. I think the prepared mind is always looking for, data that reinforces our hypotheses. And I think humans are unbelievably good at pattern recognition, but to give ourselves the best possible chance, we'll need to make sure that we're using the best possible tools and methods. So with that, I'd be happy to answer any questions that you might have. So I've opened up the chat window. If you have any questions at all, feel free to go ahead and type them there. If you prefer, you can also send me an email, and I can answer those personally without any problem.
Hello, Aaron.
Yes, hi.
Hi there. So it doesn't look like we're getting any questions from the audience, does it? Do you want to hold on a little bit longer?
Yeah, I'm happy to wait for a few.
Just for everybody to know, if you have any questions, please type it into the chat pane on your webinar. Okay, yeah, we've got a question here. So this is from one of the attendees, Colette Short. Is the ICC the same whether a microbe is aerobic or anaerobic, Aaron?
Yeah, so what the ICC does is it takes a look at all of the microbes in a sample. And what we do is we compare, who is there and how many are there at time zero, that as close to ground truth state as possible. And then we take a look at that same sample or a matched pair of that sample a little while later. And ICC is just a metric. It's sort of like a correlation coefficient that allows us to determine how similar the community composition at that later time is to that idealized ground truth state. In this case, the bigger the ICC, the higher the correlation or the better the stabilization is. So it's not about whether it's anaerobic or aerobic, it's about that entire community and how each and every member changes or doesn't change over time.
Okay, great. So we have two more questions as well. So I'll go with the first one. This is from Emily Hutt, and she says, I would like to know if you also offer help with microbiome analysis, specifically shotgun metagenomic.
Yeah, absolutely. So we have quite an interesting tool set. We're able to take a look at that with publicly available reference databases as well as things that we've curated in-house. We can take a look at the sequencing information as well as the the biostatistics behind that. So I'd be happy to answer any specific questions that you had on that. You can just send me an email for sure.
Okay, and then we have one from Rami Aziz, and this is, he thinks he may have missed this, but do you have data on the impact of the first few minutes, hours from stool collection to storage on data quality and microbial community distribution?
That's a really good question. We've tried to go as granular as possible in our stabilization studies. When we think about minutes and hours, you would need a liquid nitrogen toilet or something like that. That's a really, really short interval. But we have gotten it to 3 hours. That's the fastest we've been able to test it. And I'd be willing to share some data with you separately in terms of the kinds of changes that we do see in unstabilized samples. But I'm happy to share that in the OmniGene buffer, even at 3 hours after stabilization, the buffer itself doesn't induce any changes in that community composition. So it's what we call a neutral stabilizer. It doesn't color the community in any particular way or tilt it in any way.
Okay, we have a question from John Malcolm Greg, and this is: what other types of microbiota sampling material are you looking at besides stool? For instance, if we were doing grabs from the small intestines or stomach in a porcine model.
Good one. We have buffers for tissue, something that's called Genotek, and the microbes that would be sort of present in villi, or in some of the folds in the small intestine, that would be interesting. We would probably need to do a little bit more analysis to see how those are stabilized. But in terms of other sample types, sputum for respiratory microbiome, saliva, gum line, and biofilms for oral microbiome, urogenital sampling, and skin are all really well covered in our product portfolio. The biopsies, we would probably be able to provide a buffer sort of on its own. That's probably the best way to answer that one.
Okay, and I have two more. So this is from Ibrahim Hamad, and it's any special pipeline, I think that is, it's for study the gut eukaryotes, fungi, for example.
Sure. Definitely 18S and ITS are in our wheelhouse. We even have, actually one of our team members did her thesis on lysing aspergillus. So she really focused on techniques to make sure that all those really hardy fungi were open and accessible to whatever downstream analysis, whether that be sequencing or what have you. So that's something that we can definitely help with.
Okay, and I think we'll cover this the last question. It's again from Emily Hertz, who asked a question earlier around the shotgun stuff. And Emily's just saying, once the samples are stabilized, are the microbial cells still viable for culturing?
Yeah, good question. I would say that it would be very difficult for us to make that claim because it depends on the bugs that you're looking at. I would strongly recommend sampling in like a reduced Carey-Blair or some kind of medium specifically for culturing in order to do that. What we've done in the past to accommodate that is create sort of a collection kit, right? So one for genomics, one for viable bugs. What we found is that stabilizing on a per analyte basis, so looking at things in as optimal way as possible, provides definitely the best result. If you're trying to stabilize 2 or 3 different analytes in the same vessel, that suboptimal stabilization results in way more noise than if you were looking at 3 aliquots of that same bulk sample. So definitely 2 tubes, one for DNA, one for culture is our recommendation.
Okay, great. Okay, well, Aaron, thank you ever so much for that really interesting presentation. As you can see here on the final slide, everybody who's watching this webinar, you've got Aaron's contact details there, so please feel free to get in touch with him. I'd just like to make a final plug for the PRI, Pharmacobiologics Research Institute, and also for our conference, Pharmacobiotics Global, which is taking part in Paris, March 14th to the 15th. You can find that on the web on pharmacobioticsevent.com. If you're interested, please feel free to get in touch via the website. And again, Aaron, thank you very much for a really interesting presentation.
Thank you, and thanks to the group.
Yeah. Okay. Bye now.
Bye-bye.